moving on to topic 6.5. Um, the first question I'd like to ask you, I'd like you guys to think about, rather, is why are our cells different? And what do I mean by that? Well, first of all, think, do all of your cells have the same DNA? And the answer to that, you guys should know this already. But yes, your cells all have the same basic DNA, the same combination of A's and T's and C's and G's throughout your body, right? But why are our cells different? And what I mean by that is like if you look at something like your heart in here, your heart cell is a lot of muscle tissue, right? A cell that's located in your heart is going to be muscle tissue, very fibrous. Um, it contracts, expands, right? It has a very specific tissue type in your heart. That's going to be very different than the cells that you'll find, let's say, in your bones that deposit bone matrix and move along and kind of build up your bones or break down your bones. Also, think of your skin, right? Your skin. What about your skin cells? They offer protection, right? They build up a protein called keratin in the exterior of your body. That's what your outer layer of skin is made of. Why are all these three types of cells so completely different if they have the same DNA? And the answer to that is gene expression and regulation of gene expression. So if you'll excuse my animation, it's a little hiccuped here. I downloaded it and had to copy it down online. But our genes are expressed differently in every cell. So let's say we have a gene along our DNA. Obviously, this would be an extremely tiny gene, not likely to be something that's small. But we typically will have something called a regulatory sequence. A regulatory sequence is something, as you might guess from this root word, that regulates things, right? Regulates what's going to be transcribed and made into protein. An example of this in eukaryotes specifically would be something called a Tata box. The T's and A's in here represent thymine and adenine. And if you remember, T's and A's are easier to pull apart because they have fewer hydrogen bonds between them than a C and G, right? Just kind of something to keep in mind when you see this. A lot of times where DNA needs to pull apart, you're going to see more A's and T's for this reason. So you might have some sort of regulatory protein, such as this Tata box binding protein. So what you'll have is it bind for any specific sequence, and then your RNA polymerase will receive the signal based on this binding of this regulatory protein. Hey, I need to bind and make an mRNA molecule, which eventually, of course, will become a specific protein. Now, what if this is let's say this gene is keratin or something like that I mentioned in the skin. How's this going to work in a cell that does not need to make keratin? It's not a skin cell. Maybe it's a muscle cell. It needs to make myosin, actin, those types of proteins. Well, you're going to notice that there's different mechanisms in which we can either turn off or turn on a gene. Turn off or turn on a gene. And we're going to be talking about a few of those, particularly in the next video. Before we get to that, though, I do want to talk about one thing, epigenetics. You guys may have heard of epigenetics before, but epigenetics is kind of another layer of information, or that's the way I like to think about it. DNA is not simply, as we used to think, the exact genetic code for everything, right? And if you think about this, don't you think people turn out differently based on their life experiences? Well, it turns out that what you experience in your outside environment can have an effect on who you are, right? On how your genes are expressed. There's two ways this happens. There's something called DNA methylation, and there's also histone modification that can occur. And essentially what this is these methyl groups, which are CH3s, right? This is a methyl group that can kind of bind to places on your DNA to either stop or start transcription, or they can bind to histone proteins to either compress them and make it difficult to read or to make it easy to read, right? So this can definitely affect, and I don't want you to get too caught up into this, but do know that the methylation of DNA can affect how DNA is expressed, even in a living, functioning adult like yourself. Your DNA is constantly being affected by the environment, right? And we've seen a little bit of this when we talked about cell signaling, and it's really no different than that. So I'm going to show you again just how different some of your cells are. If you look at this, here's some. Go ahead and take a guess if you want to pause. Tell me what type of tissue this is, if you can. Tell me what type of tissue this is, and tell me what type of tissue you think this is. Go ahead and pause and tell me what you think. Okay, you should be back. This here, you might notice this is nervous tissue. You should see these kind of thin branching neurons, right? This is nervous tissue. Muscle tissue. This is muscle tissue up here. You can tell because it has these striations or stripes in it, right? And if you take anatomy, we'll be looking at this as well. This over here is skin tissue. This is skin tissue. This is the top layer of skin up here. And this line here kind of indicates the lowest layer of the skin tissue in which these cells divide, migrate up, and are eventually shed off, right? So we have nervous, muscle, skin tissue all look completely different, all act completely different, and that is because of gene expression. So muscle cells might express a lot of one gene, and skin cells may not express it at all. Nerve cells might express it a little bit, right? Skin cells this might be a keratin gene right here, this specific gene. Skin cells express it exclusively. Nerve, skin, and muscle cells all express a certain gene over here, right? So this just goes to show you how different cells produce different genes, and it is all based on a regulation process, right? And of course, this regulation is directly related to cell signaling. So keep that in mind. These cells are receiving signals from other cells, which are regulating what DNA they are deciding to transcribe and translate. Okay, so that is it for regulation of gene expression. Join me in the next video. Thank you.